Friends, welcome to my channel, and today in this video we are going to study about the overview of the immune system. Immune system have evolved to protect multicellular organisms from pathogens or harmful germs. The fully functional immune system involves so many organs, molecules, cells and pathways. The discipline of immunology come into play by the observation that individuals who had recovered from certain infectious diseases were thereafter protected from the disease. And one such experiment was conducted by Edward Jenner. The experiment was Sarah Nelms, a milkmaid, got infected with cowpox. So from cowpox, from this infected lady has been transferred to a boy named James Phipps. And it was observed that Phipps falls ill with the mild case of cowpox. Now, second time, when a smallpox infected scabs are inoculated into Phipps, this time it was observed that Phipps is unaffected. That means, first time, Phipps becomes mild infected with the cowpox. But when the second time smallpox infection was used for inoculation, the Phipps remains unaffected. That means the disease itself somehow play a role in the protection from the same disease when the second encounter is happening. So from here, the concept of vaccination started, and hence Edward Jenner, who performed this smallpox study in 1978, known as the father of vaccination. After Edward Jenner, Louis Pasteur also performed one experiment and got an observation. Chickens infected with cholera, what he did is he isolated this cholera pathogen from these chickens and grow this pathogen or bacterium in the culture. After some time, he used this bacterium culture to infect some fresh chickens, and the observation was the chickens became ill, but they recovered. Again, next time he used fresh bacterial culture. This first bacterial culture he used after some time. That means there is a time gap between the culture inoculation and injection into the chickens. Second time, he used fresh bacterial culture and injected into these green chickens, those who are previously injected, and some fresh chickens. The observation was the previously injected chicken survived this injection, whereas the fresh chicken died. So from this experiment, what it was concluded? Aging had weakened the virulence of the pathogen because first time what he did is he injected the bacteria after some time into the fresh chicken. That means there is a time gap. So during this time gap, aging happened, and because of this aging, this virulency of the pathogen got weakened, and that such weakened strains are only known as attenuated strains, could be administered to provide immunity against the disease. He called this attenuated strain a vaccine. So whenever any pathogens are attenuated, that means they made weak for injection into any fresh organism. Because of that attenuation, the virulence capacity of that strain get weakened, and due to which they don't cause the disease to a level that the fresh organism can die. So because of this, these chickens became ill, but they recovered. Second time, when the fresh culture was used, because these green chickens were previously injected with the bacteria, they survived, whereas the fresh chicken died. So this was the experiment and the observation of Louis Pasteur. Formed an experiment in a ship with anthrax. What he did was use the bacterium that cause anthrax, that is Bacillus anthracis. And the bacterium was heat attenuated. That means with the help of high temperature, the virulence capacity of this bacterium has been reduced and used to vaccinate the ships. Now, next time, a virulent strain was used. First time, the strain was not virulent because the virulency had been reduced because of this high temperature. Second time, when the virulent strain was used on the previously inoculated ships and the fresh ships. The observation was previously inoculated were survived, whereas the fresh ships, they died. So the conclusion was we can use the weakened pathogens to provide protection against the virulent pathogens. The experiments of Louis Pasteur on anthrax, on the fresh chicken, and the Edward Jenner. These experiments marked the beginnings of the discipline of immunology. Now, immunity involves both humoral and cellular component. Cellular components means cells. Humoral component means any soluble components of the blood serum. So these are mainly antibodies, or we also call them as immunoglobulins. Because these antibodies were contained in body fluids, body means humor. Why these are known as humoral immunity? Because the word humor means body. So these are present in our body fluids and provides protection against the pathogen. Hence, such immunities are known as humoral immunity. Immunity can be of two types, adaptive immunity and innate immunity. Innate means by birth. If we carry any immunity from our birth, we call it as an innate immunity. Adaptive means which we adapt during our lifetime. So that is known as adaptive immunity. Now, adaptive immunity can be natural or can be artificial. Artificial means when we purposely induce any immunity, that we call it as an artificial immunity. Natural means it occurs naturally without any interference of human. Now, the natural and the artificial both can be passive and active. Passive and active, the difference is when our body produce any antibodies for our protection, we call it as an active. When antibody is produced in some other organism's body and that antibody get introduced in our body, we call it as a passive. Passive and active. Active means when our body produce antibody for protection of our body. That is active. Passive means when the antibody is produced in any other organism's body, from that organism's body, when that antibody is extracted and introduced in our body for our protection, we call it as a passive. Now, under natural, the passive is maternal antibody. When a fetus is present in any mother's womb, the mother's antibody provide protection to the fetus as well. That is known as passive because the antibody is present in the mother's body. Clear? Even after the birth, when the baby is dependent on mother's milk. So from mother's milk, they get antibody and that antibody gives them protection. So both are mother's antibody that are getting transferred into the fetus or the baby. So this is passive. Naturally, when any organism get exposed to any pathogens and that pathogens somehow provides protection for the second attack. So that we call it as an active infection. Artificial is also passive and active. Passive is antibody transfer. Generally, this antivenom, it comes under passive artificial. Why artificial? Because it is made due to human interference. Because of any human interference, this antibody has been prepared. So antivenom. How antivenom is made? Against any venom, this venom is introduced into horse body in slow doses, due to which the horse body produce antivenom. That is antibody against venom. So this antivenom is given to us when we get any snake bite. So this is passive mode. Active mode is vaccines. Vaccine means, for example, polio vaccines, etc. So that means the antigen or this pathogen is introduced in our body only and our body produce the antibody. Passive and active. Active means when our body only produce antibody for protection. That is active. Passive means the antibody is produced in some another organism's body and that is getting introduced in our body for protection. That is passive. Natural passive is when a fetus get antibody from mother. Active, when a baby during its lifetime gain any pathogens and because of that pathogens getting protection for the second time attack. is called as active. Artificial means with the human interference. So passive means these are mainly the antivenom. How it is made? The venom is introduced into the horse body. Because of that, the horse body produce the antivenom, and this antivenom is used when any person get a snake bite. Active, when directly the pathogen is getting introduced in our body, for example, polio vaccines, etc. So for that, what happens is the polio pathogen is only getting introduced in our body, and our body produced antibody against that polio. Clear? So this is active immunization. Next is cell-mediated immunity. Cell-mediated immunity means when the cells are involved. So there are varieties of cell in our body that helps to fight against antigens. So they can be macrophages, there can be T cells. T cells can again be of T helper cell, T killer cell. Killer cell will kill the cell. Helper cell will help B cell. B cell can be again of effector and memory B cell. Memory B cell will remember the antigens for the next time attack. Effector B cell will produce the antibodies and help to fight with the antigen on the primary attack itself. So there are varieties of cell in our body which we will study in the subsequent videos, that what are the cells and how they play an important role in fighting against any pathogens or antigens. Now, how these foreign substances get recognized by our immune system, how our immune system discriminate between self and non-self. So how our body decide that this is a pathogen, this is the thing that came from outside and this is not my part. So that can be done by the presence of receptor on the immune cells. In our immune cells, varieties of different receptors are present that recognize the non-self part or the antigen or the pathogen. So the pathogen also contains some markers that are get recognized by our body or by the receptor presence on our immune cells. So it can be through humoral mode by antibody, or it can be through cellular mode, that means different cells that involve in our immune systems. Clear? So our body is very capable of discriminating between self and non-self. So because of that, it can easily able to recognize the non-self and subsequent killing of the non-self or the pathogen part. So we will see in the subsequent videos that what are these receptors and how they recognize the antigens or the pathogens that enter in our body. So this is a general overview that whenever any antigen or pathogen enters in our body, the macrophages are there that can recognize the antigen. After recognizing the antigen, they will engulf the antigen, they will digest the antigen, and some part of the antigen is presented to T lymphocyte by these macrophages only. After the encounter with the antigen, the T lymphocytes become activated and they divide into killer cell, or they can also activate the B cells. Killer cells will kill the cells that got infected. Now after killing, this can get recognized by macrophages for final digestion, or sometimes macrophages, because of macrophages, the T cell recognize the antigen and kill the cell. B cell will release the antibodies. Antibody will bind with the antigens, and this antibody antigen complexes can also be get recognized by phagocytes and they can engulf it. And also for the subsequent attacks, our body form memory cells. That means they remember these pathogens. Once any pathogen enter inside our body, they get remembered by our body cell for lifelong. They will remember this. So for the second time when the same pathogen enter in our body can fight with double power. So the killing becomes more efficient in subsequent infection by the same pathogens. So this is how our body fights and protects ourselves from the foreign substances or pathogens, or we also call them as antigens. Now one question from my end for you all. What kind of immunity the corona vaccine provides? Is it innate or adaptive? If it is innate or adaptive, then you have to tell, is it a natural or artificial immunity? Under natural or artificial, then you have to classify them as, is it an active or passive mode of immunity? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.